Hello, this is part 2 of our genetics and heredity lecture discussion. We're picking up from our last discussion talking about the ratios that we see in offspring based on the alleles that the parents can give in sperm or egg. So remember the Punnett square, we put the egg possibilities, the alleles and the eggs across one side, either horizontally or vertically. It doesn't matter which, as long as both are represented. So if this is the male, if all his cells are big E, little e, when he makes sperm, as a result of meiosis, the sperm will get either big E or little e. And that's what we put along the side. So half of the sperm would have big E, the other half of the sperm would have little e. And for a female who is big E, little e, half of her eggs would be big E and half of her eggs would be little e. So then we just combine these two to find what the offspring would be. If, for example, if this egg with the E was fertilized by this sperm with the big E, we would have an individual that is big E, big E. And when we're looking at the allele for earlobes, unattached earlobes are dominant, which is big E. Attached earlobes are recessive, so we give that the little e. So if we look at what attached earlobes look like, it means that there's no little fleshy region here that hangs down. So you can see here, unattached earlobes, there's a fleshy region that hangs down away from the side of the head. But because each of these individuals carries a recessive allele, if that recessive allele in egg and sperm come together at fertilization, we're going to end up with an individual that is little e, and we're going to see the attached earlobes. So this individual, if you look at yourself in the mirror, for example, if you have attached earlobes, that means you are recessive, homozygous recessive for that trait, and you're going to see that kind of horizontal straight line at the end of the earlobe going right into the side of the head. So that's an unattached earlobe. I'm sorry, it's attached. Sorry. Unattached means it's hanging down freely and not attached to the side of the head. So this is the attached earlobe. And it's interesting because some of our kids have unattached earlobes and some have attached earlobes. And my husband has attached earlobes. So that means I must be heterozygous, because if he has attached earlobes, that means he looks like this, and he can only provide little e to his offspring. But because some of my kids have unattached earlobes, that means I must have the dominant allele as well as the recessive allele for attached earlobes. So some of my kids look like this and have unattached earlobes. So what we can do when we have an individual that we don't know their genotype, we can do what we call a test cross. And a test cross is always looking at an individual by crossing it with someone with a known genotype. For example, if we know that someone carries the dominant trait, we know they could be homozygous dominant or heterozygous. So we don't know which they are because we can't see someone's genotype, but we can see their phenotype. We can say we know that they are a dominant, exhibiting the dominant trait, because we can physically see it, but we don't know what their genotype is. So we can perform what we call a test cross. So in a test cross, if the individual is homozygous dominant, you're going to see all tall offspring in that F1 generation. However, if they are heterozygous, then we are going to see that, like in this case, we're going to see if this individual is heterozygous, we're going to see some offspring that are going to be exhibiting the recessive trait. So we always will cross an individual with a homozygous recessive genotype. So we're going to use that individual to figure out what my unknown individual is. So we can figure out if they're homozygous dominant or they're heterozygous by crossing them with someone who is homozygous recessive for that trait. And if we see the recessive traits show up in the offspring, then we know that that individual was heterozygous. Because again, anytime we cross someone with that is homozygous dominant, every offspring is going to inherit a capital E. If this was big E, every offspring is going to inherit that big E, and they're all going to have the dominant trait as their phenotype. But if we ever see the recessive phenotype appear in the F1 generation, then we know that the parents each had little e. and if we know that one parent was homozygous recessive, little e, the only way to get this would mean that the other parent had to be heterozygous and could not be homozygous dominant. So this is an example of a test cross, and this is the individual that we know because it has the recessive phenotype, therefore it has to be little t. We're going to cross it with the individual whose genotype we don't know, and if we get all dominant offspring, then we know that that unknown parent was big T. So looking at the laws of Mendel and what he found with pea plants, we know that there are certain genetic disorders that are inherited in a certain pattern. And there's a number of genetic disorders in humans that follow a specific inheritance pattern. So some of these genetic disorders are the result of changes in chromosomes that are not the sex chromosome. So autosomes are chromosomes 1 through 22 in humans that are not the sex cells. They don't determine gender. They are what we call body cells. They are autosomes. They are not the sex chromosome. So autosomes, if you have a dominant disorder, if a disorder is autosomal dominant, it means that anybody with a dominant allele is going to have the disorder. And if they don't have the capital letter allele, then they don't have the disorder. And these are very serious disorders because it's much more likely for someone to receive the capital A when you have individuals that could be homozygous dominant or heterozygous. So these are much more common in families that carry the dominant disorder. And an example of a dominant disorder is Huntington's disease. Huntington's disease is a very serious nervous system disorder that strikes people in early adulthood before they've had children in some cases. And if they inherit that dominant allele for Huntington's, then they will have Huntington's disease. So it's a very debilitating disease that causes death. And I have a link in our folder on YouTube with a short video clip of what Huntington's disease is. You can watch that video clip. A lot of other genetic disorders are autosomal recessive, and that means that a person has to have both lowercase letters to have the disorder. And the difficult thing about this is heterozygous individuals are carriers, and they don't know that they're carriers because they don't have the disorder. So if you look at something like cystic fibrosis, for example, that is a condition that is autosomal recessive. So if I look at something like this, let's say we're looking at cystic fibrosis here, and we have two parents that are normal, but they're heterozygous for cystic fibrosis, which means they carry the cystic fibrosis allele. Let's say it was little e here in this case. Let's say that was cystic fibrosis. Half their eggs and half their sperm contain the bad allele. So if this allele and this allele come together at fertilization, you're going to have a child with cystic fibrosis. So anybody that is homozygous recessive is going to have the disorder. So cystic fibrosis is a very serious disease. It affects the lungs. It causes excess mucus production throughout the body, and particularly the airways in the lungs are affected, and those individuals have lifelong health problems. They're very much at risk for death when they get a simple respiratory illness. And I've taken care of some young people, young college-age kids. I've had some in my classes even that are suffering with this disease, and one of them has passed away that I know of for sure. And it's just a really, really serious lifelong problem. And some people don't make it to 30 years old because of their lungs becoming so infected. They need either a lung transplant along with a liver transplant because of all the antibiotics and medicines they have to take to keep their lungs healthy and functional. Has a really hard impact on the liver, so they end up having liver disease as well as lung disease. So, looking at how an autosomal recessive disorder would be inherited, if we look at something called a pedigree, what a pedigree is it shows the parents and their offspring. For example, the circles represent females, the squares represent the males. So if we have a male that we do not know what his genotype is, we can see how things can be inherited across generations. So these two people had children. So the horizontal line means that they got together and had offspring. So if we look at what offspring they had, follow the vertical line down, they had two female, so they had two daughters that were heterozygous. So that means that they inherited an allele, a recessive allele from a parent and a dominant allele from a parent. Well, we know for sure that the dominant allele came from this parent, from the father, because the mother did not have the dominant allele. So now when they, again, find someone and have offspring, so in this case, this female had offspring with this male, again by a square, and they had 3 children, 2 sons and a daughter. And then we look at these children, a daughter gets together with a male, has a daughter, that's their generation. So this is the second generation resulting from these parents. This is the third generation. So here we can see, we look at, this is interesting, right? Because we have, it looks like this would be cousins, a female having a child with this male, and then here comes that recessive allele. But again, this is possible because we have a recessive allele here and a recessive allele here. So if those were present in sperm and egg, that could come together and form children with the homozygous recessive genotype, meaning they have the disease. So when we shade in the circles or squares, it means they have the disease. So now we can figure out, what do we think this individual is? Well, this individual could either be heterozygous or he could be homozygous dominant, because we don't know which allele this individual inherited. So there's a lot of unknowns here that we can look at because we don't know how things were inherited here. So, but pedigrees give us a little bit of a clue as to what possible alleles have been inherited. So they look at this with quite a bit of scrutiny to determine who has the disease and who doesn't, and what possible alleles can be. So again, the shaded squares and circles mean that they are affected and they have the disease, and some might be carriers, so we don't know if they have the allele or not. So here's an autosomal dominant pedigree. This would be like Huntington's disease. So we have 2 individuals, they have the dominant allele, so they are affected. So again, autosomal dominant diseases means if the dominant allele is there, whether they're heterozygous or homozygous dominant, they have the disease. So these 2 individuals have Huntington's. It may not have developed until they were in their 30s and they had these 4 children. So we can see there's 4 kids here. And with these individuals, they inherited the big A, so they have Huntington's. This person had a child with a person who did not have Huntington's. Some of their kids inherited that dominant allele and some didn't. The good news about autosomal dominant disorders is that if you did not inherit the allele, it's gone from your pedigree. So if the little a and the little a came together to form these 2 daughters, they will not have Huntington's and they will not pass it on to their children because they don't have that dominant allele. So this side of the family is clear. Nobody's going to have Huntington's and it's been wiped out because nobody inherited the dominant allele. this side of the family is going to continue to pass Huntington's along if they continue to have children because they inherited that dominant allele. And this is interesting. This was in my family. 3 generations ago, Huntington's was on my mother's side. But my grandmother did not inherit the allele, so then she couldn't pass it on to her children, and our family is free of this disease. So autosomal dominant diseases, are very serious. They're, when we have people inheriting the disease shows up more frequently because that dominant allele can be heterozygous or homozygous dominant and cause disease. So again, cystic fibrosis, I said, was autosomal recessive disease. It causes a defective channel for chloride, which causes a salt imbalance and results in a really thick mucus. And when we have thick mucus in our lungs, we can't clear our lungs. Bacteria will settle in that mucus, and people need lots of breathing treatments. This is a percussion vest, which when you plug it in, it causes the whole body to shake a little bit, and it shakes the mucus loose in the lungs, and then the individuals will cough up that mucus after their percussion therapy vest session. So like I said, I have students that have had this disease, and I've had patients with the disease as well, and it's a very serious, debilitating disorder. So this is just, shows an example of how Huntington's is passed down. So here's the allele from Dad. We can see that they had 2 daughters and 2 sons. 1 son and 1 daughter received the allele, so they have the disease, and they can also pass it down to half of their kids, or half of their sperm and egg, I should say, because half of their alleles are the defective allele. And these children did not inherit the allele, and they cannot pass it on to their children. So some traits are controlled by multiple alleles, which means it's not just one allele that we can follow through a pedigree using a Punnett square. For example, our blood types. We have blood type A, we have blood type B, and then we have individuals that have neither blood type A or B, so we call them blood type O. So these, there's 3 different alleles that someone can inherit, and A and B are equally dominant. So if someone inherits the A allele and the B allele, they are type AB. So we call that codominance. Codominance means 2 are equally dominant. So A and B are equally dominant, and blood type O means you have neither A or B alleles. So we say that you are blood type O. So a person can be heterozygous for blood type A, which means they have both A alleles, or they could have the A allele and the absent allele, or the O allele. So homozygous dominant, heterozygous. The same thing with B. You can be homozygous dominant or heterozygous. But if you inherit A and B from a parent, we say that you are blood type AB. This is not considered heterozygous because B is not recessive. We say this is codominant. Both are equally dominant, and you are blood type AB. And this individual came probably from heterozygous parents or from O parents, and all they inherited was the little I and the little I, which makes them blood type O. So incomplete dominance is another type of dominance. So we talked about codominance, means they're equally expressed. So blood type AB is an example of codominance. Incomplete dominance means the heterozygous individual is somewhere in between the homozygous dominant and homozygous recessive. So if homozygous recessive is white and homozygous dominant is red, in between red and white is pink. So the heterozygous individual would be pink. Another example of this would be if you had, say, a rabbit that black was dominant, white was recessive. Incomplete dominance, what color would the rabbit be? If you answered gray, that is correct. If that inheritance was codominant and you had a black rabbit and a white rabbit and they were equally expressed, the heterozygous individual would be black and white spots or black and white patches. So codominance, they're equally expressed. Incomplete dominance means there's a middle ground for that heterozygous individual. So here's an example. We have a pink flower, and when we cross 2 pink flowers, incomplete dominance, they're looking at R2, I don't like the way this is expressed with the R1 and R2. I think that's confusing. But R1 is dominant and R2 is recessive. So these are two heterozygous individuals. So the R1 they're considering dominant, the R2 is considered a recessive allele. I don't like how that's expressed. That's not consistent with the way we've been using our Punnett squares. But if R1 is dominant, R2 is recessive. Two R1s makes a red. The heterozygous person is pink, or flower is pink, and the homozygous recessive flower is white. So that's incomplete dominance. So examples of incomplete dominance that we see for humans is people that have cholesterol issues. Sometimes we see people that have problems with cholesterol problems early in life. This is called familial hypercholesterolemia, which means high blood cholesterol. So if you are homozygous for this disorder, homozygous recessive, then they have serious problems and heart attacks in childhood. If they're heterozygous, they have less of an issue with cholesterol, but somewhat of an issue. So they have heart attacks later, in life, early adulthood. And homozygous dominants don't have the disorder at all. So that's incomplete dominance. Another term is incomplete penetrance. This is when you have the allele, but it's not actually exhibited. We don't see it in the individual. So, for example, polydactyly is having extra fingers and toes. And some people will inherit the allele, but they won't actually have the trait. So that's something called incomplete penetrance. Think of penetrate. It doesn't penetrate into the phenotype, even though they have the dysfunctional genotype. Pleiotropy is when we have a certain set of alleles that have a phenotype that has specific traits, like you have one single allele, but the traits are exhibited in a bunch of different areas. For example, Marfan syndrome is the result of a single allele, but it causes changes throughout the body, not just attached earlobes or a widow's peak, not just one specific trait, but many traits. So think of plio as many traits from one single allele that is dysfunctional. So Marfan syndrome has causes a lot of problems with connective tissue. So and it causes abnormal growth. So people have long arms, hands, legs, and feet. They have a weak aorta. So that big blood vessel that carries blood out of the heart to the rest of the body is weakened because of the connective tissue damage, and they have poor eyesight. So that is just, sometimes they can have learning disabilities, but not always. It's actually a pretty common syndrome. I know a couple of people that have this syndrome, and they're highly functional, intelligent individuals. So, but they do have some of these other characteristics. So that's pleiotropy. Another concept related to genetics in humans is polygenic inheritance. Sometimes we see alleles that are carried on two or more chromosomes, and they result in a variety of phenotypes with a variety of characteristics. For example, human skin color. Some people can be very dark skinned, medium skinned, light skinned. Those are controlled by alleles on multiple chromosomes. Height. We see, parents of a certain height, and they have 6 children like myself that are varying in height. So a lot of differences in height because those genes for height are across different chromosomes and have many alleles. Eye color is another example of polygenic inheritance. Poly meaning many genes control these characteristics or traits. Another concept is X-linked inheritance, which means these traits are carried on the X chromosome. So if it's carried on the X chromosome and females, give X chromosomes to their offspring, and males can give either X or Y to their chromosomes. Y is what determines if someone's going to be male. So if a bad characteristics or a disease is carried on an X chromosome and a mother has the disease and it's homozygous recessive, that means both of her X's are bad X's that carry the bad allele for that X-linked trait. Then all of her sons are going to have that trait because she can only donate a bad X to the males, and the males don't have another X to kind of like a heterozygous individual don't have a good X to mask the bad X. So what we see in these individuals, I'm going to make this a little bigger here. Color blindness, what we find is if a woman is colorblind, she donates that bad X to her son, and her son has an X and a Y. The Y comes from the dad, the X comes from the mom, and her sons are going to be colorblind. Another one is Menkes syndrome. This causes issues with how we manage copper in our environment and in our cells, and this causes a lot of widespread problems in individuals that have this syndrome. Muscular dystrophy is something that we see the Jerry Lewis telethon every year, raising funds for these kids, but their muscles break down essentially, and these people don't commonly live past 20 to 25 years old because of muscular dystrophy. That's carried on the X chromosome, and oftentimes mothers can be carriers of muscular dystrophy, so they have a good X and a bad X, and if they donate the bad X in that egg during meiosis, and that is what is fertilized by sperm during fertilization, then that son is going to have muscular dystrophy. Daughters can have it as well, but it's less common. Another one is adrenoleukodystrophy. This is a disease related to the adrenal gland and causes problems with, I'm not even familiar with that one. I'll put a YouTube link on that one in our Blackboard folder because that's not something I've ever seen in patients in the hospital, so it's probably not as common. Hemophilia is one that deals with blood bleeding. It's a bleeding disorder. So if someone inherits the bad X that carries the trait for hemophilia, then they are going to have a problem with bleeding. So the biggest thing to note here is if a mother has the bad X allele, and that is what is fertilized by sperm carrying the Y chromosome, all males that are XY are going to have the disease. So here we can see a grandfather that has the bad X. Let's say this is hemophilia, or let's say this is color blindness. So when they have children, here we have a daughter and a son. The daughter inherited the bad X, but because she had the good X, she does not have the disease. She's just a carrier. And then if she has children with a male that has the good X, then the chance of any child getting the bad X is much lower, because we can see here that all the children inherited the good X and therefore did not have the disease, but this child got the bad X from the mother and the Y from the father. So this son, this grandson, inherited the disease from the grandfather. Same thing here. We look and see that this son of this pair, this partnership, this son married or had children, I have to say married, but had children with an affected female, then the chances of the sons getting that disease is 100%, because they can only donate the bad X to the son, and as a result, every son will be affected. So again, that's the rule, and you need to know this for our test, is anytime you have an affected female with a disease, an X-linked disease, every son is going to also have the disease, because she can only give bad X's to her sons, because the father can only give Y for it to be a son, and therefore every son will be affected. So here's just a picture of what muscular dystrophy looks like. Again, these children have very thin arms and legs because their muscles essentially waste away. So that is the end of our genetics and heredity unit. Be sure you go through the Learn Smart homework and the test, and our next topic will be viruses, bacteria.